Assalamualaikum and a very good evening to Madam viewers. In today's video, me and my group 6 from AS1204C will present to you about chromatography analysis in creating vaccines. But before we start the video, let me introduce you to my fellow group members. So, this is my fellow group members. My name is Shiman bin Masdi, and we have Karil Najib here and Aung Shahmi Shairan. And without further ado, let's start with Karil's part.
Before we begin, I should introduce what is chromatography analysis. Chromatography analysis is a technique used to separate and identify components within a mixture based on their differing affinities to a mobile phase and a stationary phase. As the mobile phase passes through the stationary phase, components of the mixture interact differently, causing them to move at different rates. These separations allow for the identification and quantification of individual components based on their retention times or elution order. But why chromatography analysis is important? Chromatography analysis is essential in vaccine manufacture because it verifies the vaccine's purity, safety, and effectiveness. It aids in the separation and removal of contaminations, ensuring the presence of the correct compounds and maintaining uniformity across batches. This method also screens for impurities and ensures the chemical, such as adjuvants and stabilizer, are properly added. Chromatography contributes to regulatory compliance by offering extensive analysis, ensuring vaccine safety for public use. First of all, a membrane chromatography-based downstream processing platform with a demonstrated industrial application potential was established. Cell culture-derived influenza virus H1N1 APR8/34 was harvested from benchtop bioreactor cultures for the clarification of the cell culture growth. A depth filtration was selected as an alternative to centrifugation. The proposed membrane chromatography purification is a scalable and generic method for the processing of different influenza strains and is a promising alternative to the current industrial purification of influenza vaccines based on the ultra-centrifugation methodologies. This study focuses on the development of a robust downstream process for the purification of pandemic influenza strains at a bioreactor scale. A two-step purification process consisting of depth filtration followed by anion exchange chromatography is proposed. The filtration proved to be a preferred alternative to centrifugation. Then different membranes including NatriFlow HQ, Cytobine Q, and Mustang Q were evaluated for the purification of the H1N1 viruses as a model of influenza pandemic strains. Successful recovery was demonstrated while establishing the operation conditions.
The next part is materials. One of the materials that we will need are bioreactors and culturing equipment, which are 1 liter and 3 liter Applicon benchtop bioreactors, shake flasks, and heating blankets. Next are cell culture media and supplements, which are serum-free high-cell trans-FX medium, L-glutamine, colifor, and cell boost 5 for fat batch operation. Third are suspension human embryonic kidney cells. Next are control reagents, which consist of carbon dioxide and sodium hydroxide for pH control. Furthermore, we will need influenza virus strains, which are H1N1, H7N9, and H3N2. Lastly are infection agents, which is TPCK trypsin at concentration of 1 until 2 microgram per milliliter, depending on subtype multiplicity of infection MOI reagents. The picture that you are watching right now is the preview for creating vaccines using chromatography analysis. The first step of the methodology is the production of cell culture and virus influenza. Suspension human embryonic kidney cells were cultivated in serum-free medium using bioreactors. The bioreactors were inoculated at cell densities between 0.3 and 0.6 million cells per milliliter. The culture was maintained at 37 degrees Celsius with a stirring speed of 100 rpm and a pH of 7.2, using carbon dioxide and sodium hydroxide for adjustments. Meanwhile, For influenza virus production, the cells were infected with H1N1, H7N9, and H3N2 strains at cell densities of 2 or 7 million cells per milliliter. To aid viral infection and replication, TPCK trypsin was added. After infection, the temperature was shifted to 35 degrees Celsius for H1N1 subtype. The second step is harvesting. For cell culture supernatant, which contains virus particles, was collected 48 to 72 hours post-infection. To remove cell debris and other particulate matter, the harvested supernatant underwent clarification using filtration. For the step of centrifugation and filtration, first, the harvested culture was centrifuged to eliminate large cell debris. Then, the supernatant was passed through a low micron filter to ensure the removal of fine particulates. The next step is carrying out membrane chromatography. The membrane chromatography unit was equilibrated with phosphate buffer. Next, the clarified virus containing supernatant was loaded onto the membrane chromatography unit, ensuring that the flow rate and loading capacity were optimized for effective virus binding. To remove any unbound proteins and contaminants, the membrane was then washed with a washing buffer. This process ensures a purified virus sample for subsequent analysis. The fifth step is carrying out concentration and buffer exchange. The eluted virus fractions was concentrated using ultrafiltration to achieve the desired concentration. Following this, buffer exchange was performed to transfer the virus into a suitable formulation and storage buffer. The concentrated virus was then sterile filtered to ensure sterility. Furthermore, quality control and characterization also need to be carried out. The virus titer was measured using hemagglutination and plug assays techniques. To analyze protein impurities, SDS-PAGE and HPLC were used. Meanwhile, electron microscopy was implemented to assess the virus purity. Lastly, the immunogenicity of the purified virus was evaluated using in vitro assays. The last step is formulation and final fill. In the final formulation and fill stage, the purified virus was combined with stabilizers to create the final vaccine product. This formulated vaccine was then carefully filled into sterile vials or syringes under strict aseptic conditions to ensure safety and purity. The completed vaccine was stored at recommended temperatures, typically between 2 to 8 degrees Celsius, until it is ready for distribution.
And as for my part, I will be talking about the results and data for the research. And as you can see here, this is the table of evolution of different membranes for ion exchange chromatography. So this is the analyzed results of optimal chromatography steps for influenza virus purification. So first, optimizing the chromatography stage for influenza virus purity. Second, membrane selection for H1N1 production using NaturFlow HDQ and Startobind Q. Higher dsDNA built up in run-3 material, showing strong benzonase treatment efficacy. Startobind Q recovered 70.7% of HA at 1 m salt concentration, which is 28.7% greater than NaturFlow HDQ. NaturFlow HDQ eluate removed 35.3% of DNA, which is 24.5% more than Startobind Q. NaturFlow HDQ was more effective for protein elimination, which are 83.8% versus 74.9%. Last but not least, Startobind Q was chosen for further purification process improvement due to its excellent HA recovery. So this is chromatography study on HA recovery, dsDNA, and protein removal. As you can see, Mustang Q and Startobind Q showed recovery rates of 54.2% and 65.3% respectively. As for Mustang Q, it had 8.2% HA and 67.3% dsDNA. For Startobind Q, had 3.0% HA and 17.2% dsDNA. To balance recovery and dsDNA removal, fractions eluted at 300 and 700 millimolar were combined. And last but not least, Mustang Q had higher protein. As for my next part, I'll be talking about uses of vaccine in real-life situations. So first one, outbreak control. Seasonal flu vaccines require careful formulation and purification to match the strains, predicted to circulate each year, ensuring they are safe and effective. The second one, routine immunizations. As for childhood vaccines, routine vaccinations protect children from diseases such as measles, mumps, rubella, diphtheria, pertussis or whooping cough, polio, and Haemophilus influenzae type B. These vaccines are usually given according to a recommended immunization schedule during early childhood. As for adult vaccines, such as the flu shot, tetanus booster, shingles, and HPV vaccines help to protect against diseases that the adults are at risk of contracting. And for the third one, pandemic control. COVID-19 vaccines have been critical in reducing the spread of the virus, decreasing severe illness and hospitalizations, and allowing for the return to normalcy in many parts of the world. And last but not least, travel protection. For yellow fever, travelers to certain regions, such as parts of America and South America, require the yellow fever vaccine to prevent infection. And for typhoid, travelers to areas with poor sanitation may need the typhoid vaccine to protect against typhoid fever. To sum up everything, the study compared first depth filtration to centrifugation while purifying influenza strains using chromatographic membranes. The results indicated that the clarifying procedure performed similarly to centrifugation. To separate influenza viruses and pollutants, 3 commercial ion exchange membranes were used, such as NutriFlow HDQ, Mustang Q, and Sartobine Q. Sartobine Q recovered more HA despite removing fewer impurities, but Mustang Q performed better in impurity removal. The optimized operating conditions were evaluated with pandemic strains H7N9 and H3N2, yielding similar downstream process performance. I hope you enjoyed watching the video, and I hope some of the information can help to increase self-knowledge and spread awareness amongst the society. And that is all from us. Thank you and goodbye.